Hi, this is Adam Kinkaya. I'm presenting an update on my PhD progress towards my computer science PhD. It is currently April 2025, and I am going to share what I've been doing over the past year. So I originally finished my computer science master's degree at FAU in the spring of 2020. And then I took some time off for a few years. I started taking some more graduate courses and I started studying for the PhD qualification exam. I was able to successfully pass the exam 1 year ago at the end of spring 2024. I did the oral exam where I was given a paper and I had a few weeks to prepare a 20-minute presentation to three different professors. They all agreed that I did a good job on the presentation and I passed the exam that way. And then last summer in 2024, I did 3 credits of an independent study with Dr. Shankar, and I'm going to share some of my work that I did. I'll be sharing that today. I did that over the summer, and then in the fall, I took my first 3 credits of PhD dissertation. So these are the only 3 credits of actual thesis dissertation that I've taken so far. So a little bit more about me. I grew up outside Orlando, and I'm currently living in Orlando again. I've lived around the country at a few different places. I lived in Cleveland, Ohio for a few summers while I was an intern there. I interned at the NASA Glenn Research Center in Cleveland. And then I spent about 4 years living in California, in the San Francisco Bay Area and Los Angeles. I originally went to UCF, where I graduated in 2008 with a bachelor's in physics. Especially, I was especially interested in astronomy at the time. I wasn't really able to get a physics job with that, so I started working as a quality engineer for Lockheed Martin. I worked on various military airplane aviation programs, inspecting parts, doing audits, and visiting suppliers. I was doing that out in California, and then I decided, all right, I want to go into computer science, get some useful skills to get some better career. So I went to online through Oregon State University. I finished my computer science bachelor's degree, and then I ended up moving back to Florida, where I got a job as a software engineer that I've been working now for 9 years. I work for a large company that handles the software at airports around the world. We have a large market share. We handle all of the software at the airport that is internal and as well as customer facing. I particularly work in the passenger processing department, which I deal with all the ticket scanners, the boarding pass scanners, facial scanners, bag tag printers, all those things you use at the airport. I primarily work with Java and then web stack technologies such as Angular and React, PHP, SQL, those kind of technologies. Primarily Java we use on the back end. We've started using Python and PowerShell scripts a lot. That's something I've been learning at work recently. And then once I started working a few years, I decided to get my master's. I came to FAU and I completed my master's in computer science in 2020. So the thesis that I've come up with um, after talking with some of my professors over the years The title we've come up with is an executable mixed level concurrent platform for biological pathways. Basically, I'm hoping to create a software platform that will take multiple inputs from multiple biological systems at the same time and is able to in a event-driven way model the body's response to these inputs. Um, and event-driven in the sense that these are multiple systems at multiple levels going on at the same time concurrently. We're talking at the genetic level, up to the cellular level, up to the entire body systems, such as the immune system, and then everywhere in between on those levels. And all of these are events are going on in very complex ways. Um, these signals, there are feedback loops, positive and negative. There are sudden changes where a signal can be steady and then a sudden on-off state can occur. There are, of course, everything is asynchronous. Um, so this is a quite complex problem. For my particular interest, I'm looking to model the immune system and the immune response, inflammation response um, of the body. So there is a language called System C that is specifically designed for these event processes. And the idea is to use the System C language as the language for this platform. Um, we think that this will be the best language for modeling these dynamics that we see in the body. And as I said, my primary focus is going to be on the immunity pathways, inflammation response of the body, because almost all modern diseases, the main diseases we see in people, all are related to the immune system. We see this throughout modern medicine. A lot of things are coming back to the immune system response. So this is a really key area for future research, future medical advances. The idea is that these pathways in the body have already been modeled out mathematically on paper with equations, and we have the actual biological details of the pathways mapped out um, online in databases. So the idea is that everything is available. We just need to take this information, put it together in a platform, and start building up the contributions from multiple levels and multiple systems to the immune response of the body. Now, at my time as a graduate student at FAU over the last 5 years, I would say that these two topics, GWAS and AI, have been the dominant topics in my classes. GWAS being the genome-wide association studies, which are studies where we attempt to relate genetic variants, SNPs, single nucleotide polymorphisms, genetic variants, we relate them to a disease state, having a disease or being protected from a disease. The idea being that we can somehow say that certain genetic variants are associated with a disease or associated with protection from a disease or associated with some other biological state. Of course, AI is the dominant topic in all of tech today. So over the last 5 years, I've taken several classes over both of these subjects. I took 2 classes with Dr. Shankar, Semantic Web Program and Human Genomics, where we looked into GWAS studies. We looked at the online tools, the online databases. Um, we went in depth into the theories of GWAS and started looking at the equations and such that I mentioned previously. And then, of course, AI. I took several AI classes at FAU over the years. I did take briefly this AI and biology course. I was not able to finish the course, but I was able to complete a lot of the assignments in it, and um, it opened up a lot of ideas for incorporating AI into some of these systems. So when I'm building this platform using System C and trying to model things um, out of the textbook and out of the database, I do want to try to incorporate AI to try to make it better, to try to make it more efficient. Um, see what I can do with that. The idea being that after I finish my PhD, hopefully, and I graduate, I'll be able to take this platform, improve it over time, increase as more and more data becomes available, add more data, add more layers to the system to hopefully improve the production over time, the efficiency. So for a PhD at FAU, it requires 18 credits of coursework beyond the master's. I've taken 12 credits so far, so I have six credits, about 2 classes left. It requires 24 credits of dissertation. I've taken 3, so I have 21 credits left. And then it requires 2 semesters of the graduate seminar. I took one last fall, and I will take another one this fall when it becomes available. So my current plan of study that I have shows that this summer I'm going to take 3 credits of dissertation. This fall I will be taking 3 credits of a class. Right now I'm looking at the natural language processing class. I'm actually kind of running out of classes at FAU that I can take. Um, if something else more interesting becomes available, I may take something else. But right now, that's what I'm looking at. And then I hope to be able to also take 3 credits of thesis, but we will see how the workload is. And then, as I said, I'll take that other graduate seminar in the fall. Same thing in the spring, 3 credits of class and hopefully 3 credits of thesis, depending upon what classes are available. And then I'm hoping to finish it off fairly quickly after that in the next summer and next fall, do a total of 12 credits of dissertation to hopefully finish by the end of fall of next year. So a year and a half from now. So this past summer, I did a independent study with Dr. Shankar. We looked at a paper that was related to bladder cancer, trying to find differentially expressed genes, DEGs, in patients that have bladder cancer versus patients who do not. There's a particular focus on the prognosis because there are different types of bladder cancers. Some are more survivable than others, and we actually can, in theory, see this in the DEGs, certain differentially expressed genes. Certain variants are associated with certain types of bladder cancer that are more survivable than others. So this original paper, we looked at it and analyzed it, and I attempted to recreate as much of the result as I could using some similar methods. The input in the paper were these mRNA expression profiles from these online databases. These were bladder tissue samples. Some tissues were cancerous, some were not. And we had mRNA profiles from these tissues labeled as cancerous, non-cancerous. The paper analyzed this data using weighted gene co-expression networks and pathway enrichment analysis. It basically took all the genetic data, put it together, and tried to see which genes were co-expressed together, as in multiple genetic variants would be expressed together in a patient that has cancer, multiple genetic variants would be expressed together in a patient who does not have cancer, trying to group the variants together because it's not just one DEG, it's not just one, necessarily one gene variant. It could be hundreds of them, the contributions, small contributions from hundreds of them. So after they did this, they identified 3,700 DEGs that are associated with either bladder cancer or not having cancer. In particular, they identified these 4 genes as particularly significant biomarkers that if you have high levels of expression of these genes, um, you could make a prediction on the survivability of the cancer, whether that's they did it in terms of years after diagnosis. So this is a quick workflow of how I attempted to reproduce some of this results. As we said, we started off with the RNA profiles from the both cancerous and normal bladder tissue samples. I used an R notebook, R language notebook, and R packages provided by BioStar to identify DEGs. So I use these packages, Lima, WGCNA, um, to find the genes with biological relevance, that is to say, the DEGs that are related to having cancer or not having cancer. Um, from that, I was able to find 76 genes from the tumor samples and then 11 from the normal samples. So these were genes that we found to be overlapping in multiple samples, highly expressed in multiple patients with multiple tissue samples. Then the idea was to use some online tools to take these approximately 87 genes, to visualize them, look into the connection that each gene has to protein, look at how they interact on a protein level. And then I didn't get this far, but in the original paper, they have what they call a survivability calculator, which they're able to, based upon the presence of certain gene variants, they predict the time of survivability for a patient in terms of years. So this is the data that I was using from online, from this TCGA bladder cancer website. Total of 412 tumors versus 19 normal samples. This is some of the R code using this GDC query tool that is provided. You pass in the different options. So, I'm looking for the RNA data. I'm looking for tumor and normal tissue. So this is the query. We run the query. You get back a set of raw gene counts for the genes, the presence of the gene in each case. So this is some of the raw gene counts you can see. So on the left here, the left column, these are the actual genes. Along the top, or I should say each row is a gene, while each column along the top is a patient. So patient 1 has 0 T in MD genes, but 21 in patient 2, for example. These are just raw gene counts, the number of times that the device measures that particular gene as being present. It's just a raw count. Now, once I had that, I was then able to use some other existing R packages, Edge R and Lima, to identify those raw counts that were most significantly expressed in the patients. So I don't want to get too much into here, but this is some different. I was following a tutorial online about how to use these R tools to identify DEGs, um, and get some results. This is a graph that shows that as the count goes up, the standard deviation goes down. Um, from what I understand, this is just showing that it is what we expect the statistical model to be. Once we then plot out these primary component analysis or principal component analysis, we're just breaking down the dimensionality so that try to visualize the genes. But this is some of the results I had. You can see that the um, normal tissues are in red, and they're generally located in this green area, while the cancerous tumors are everywhere else. So this is basically telling us that there is a possible relationship genetically with the gene variants that differentiates tissue that is non-cancerous from cancerous. If there's not a complete separation, we would hope to see a complete geometric separation here, like a straight line dividing the black and the red dots. We don't see that, but we do see that the red dots are in a confined area. And then this is the volcano plot. You can see um, some are quote protective DEGs, as in if you have them, you are less likely to have cancer versus those that are more likely to have cancer. We're able to see the p-value here, the significance of each one, and we're able to see a clear differentiation here. This log fold change shows the either or negative contribution, so either cancer or protective or non-cancerous. And then real quickly here is a heat map. That's another way of demonstrating the differentiation between the two sets of genes. These genes and these on the top and bottom, blue and orange, you can see some are protective, some are indicating possibly related to cancer. These graphics on the top and the side are attempts to group together the genes so that see the co-expression, try to simplify the contributions. So instead of having to look at hundreds of multiple genes, we can group them together into groups, modules, is what in the original paper they call. This is sort of where I stopped on my particular work, but in the paper, they put them into modules, go into the protein analysis, protein interactions, and then get into the survivability calculations. So as I said, I have found 86 DEGs. After filtering for those with the most significant contributions, there was only 12, 6 up, 6 down. When you go online and look into the Go Ontology website, you can sort of see some biological functions, pathways related to these things, possibly related to cancer or bladder cancer. So we can say, you know, these genes are possibly, these pathways are possibly contributing to the development of tumors. The results I got are actually not that significant in that the adjusted p-value is too high, but it's it's something. And there is some overlap in my 12 results and the original paper. Multiple of these genes are in the original paper's results as well. So I am getting some consistency, at least in my methods of identifying DEGs. And here are some of these uh, biological functions, pathways associated with these DEGs. So, and then after finishing that in the fall of last year, I just started trying to learn some of the biological background that I will need. I took a series of 4 courses through Coursera and the Mount Sinai Systems Biology specialization, where I learned about a lot of the things that I had previously learned from Dr. Shankar's classes, but I learned them more from a software perspective. I actually started using MATLAB to model some of these cell cycles, like these bistability systems. As I mentioned earlier, you can have a system that's stable, and then suddenly a small input sets it over the edge, flips it on and off, those kind of things. So I spent a lot of time on that. And then I also started learning this System C language. There's some tutorials online. There's a user group online of System C people. I started just some basic getting it up and running on my laptop. Started on the tutorials and following some textbook and YouTube series. So my next steps this summer, as I said, I'm hoping to take 3 credits of dissertation. I'm going to continue learning the System C and hopefully start actually building some simple platforms. And then I'm hoping to finish up my bladder cancer work from last summer and turn it into at least a conference paper to show that I was able to replicate some of the original authors' results using different methods. And then I will hopefully submit that for publication for a conference. And then I said this fall I'm hoping to take 3 credits of a class, whatever is available, and then graduate seminar, hopefully another 3 credits of dissertation, where I will continue to work on the platform and then, as I said, submit that paper for publication. So that is where I stand today. I know my progress is just getting started, but I'm hoping over the next year or two to be able to dedicate more time and make some more significant progress. So if there's any questions at all, please contact me, email me. I can respond over email, or I can record some video responses. So thank you, everybody, for your time, and I appreciate it.